composed of just billions of individual cells. Talk about the brain. People discuss the brain as an organ. Don't forget, the brain is composed of billions of individual neurons. And the individual neurons, the chemistry of the neurons is the same, all right? Now, the proteins, the neurotransmitters that they use to communicate with each other are different depending on the class of the neuron. But the overall chemistry of the neurons is the same. So groups of neurons are classified based on whether they're more close to the brain, the central nervous system, versus the peripheral nervous system. But both of these systems are simply composed of cells that look like the sticky hand that you can buy at the drugstore. And the information comes in on the end of the dendrite and propagates down the axon, and then that information leaves the terminal. And then the addition of myelin sheaths only on the axon, myelin sheathing only occurs on the axon, increases the rate that the action potential or the nerve impulse moves or propagates along the plasma membrane of the cell. Okay. That's a really important comment, that the impulse actually moves along the membrane. When educators put this wiggly line across the cell, students start to think that the impulse moves through the cytoplasm, and it's really hard to draw in only 2 dimensions. But what really happens here is the action potential bounces along the surface of the dendrite like such. Hop, 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 before it finally gets to the axon, and then it really goes across quickly. And so this is the wave that we were talking about when you're in the gym and everyone's doing the wave in the bleachers, and you see the wave going around the gym. I see that's an action potential, all right? So how does that work? As the different areas of the axon depolarize with the flow of cations, specifically sodium, that causes this area of the neuron to depolarize as the anions flow into the cell. And that causes the next area to open the channels and depolarize, which causes the next area to open the channels and depolarize. And so it's depolarize, 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 depolarize. So as these different areas depolarize, the nerve impulse or the action potential moves or propagates along the axon. Just like when you're sitting in the bleachers and you see the wave coming, right? And the person next to you stands up, then that stimulates you, boom, to stand up. That's exactly what the sodium channels do, all right? Sodium channels are closed. Once the channels next to that channel open, that causes these channels to open, which causes those channels to open. And so it's open, 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 and the depolarization moves along the membrane. This depolarization is possible because of the work of the sodium-potassium pump. We talk a lot about the negative charge on neurons, and so students assume that there's a negative ion, an anion, like chlorine, okay? There's no negative ion here at all. It's actually the disproportionate transport. What does that mean? It means that there's an unequal transport of sodium out of the neuron versus the number of potassium in. So there's 3 sodiums out and there's 2 potassiums in. This imbalance in cation transport causes the inside of the neuron to gather what we call a net negative charge. There's no actual negative ion here. Okay, there's no anion. It's the accumulation of a debt. This is actually charged debt because there's 3 positives going out and only 2 positives coming in. So you then have this charge across the membrane. You have the positive charge outside and then the negative charge inside. So you have the potential for movement, okay? But the sodium can't move because the channels are closed. Thus, we say there is a membrane potential across the neuron plasma membrane. There's the potential for movement of sodium, but the sodium can't move because the sodium channels are closed. Okay? Until those sodium channels open when they're stimulated, and then holy cow, then there's a ton of movement. So the image after that, don't forget these green lines. This is the plasma membrane of the neuron. What you're looking at here is the membrane of the axon specifically, Okay, so here's the sodium potassium pumping the 3 sodium out and 2 potassium in. Tie this back to that core 1 question. Where's that core 1 question? There it is. Back in core 1. I don't know why they bury it so long ago. I wish they would either bring this content closer, but they don't. So, okay. So go back to core 1 and grab that 4C question. That's important. All right. So the sodium channels are closed. That's why there is the potential for movement. Okay. Then there is stimulation. There's a lot of chemistry here that we're not getting into now. So I can just casually say there's stimulation and you'll get the chemistry later. Again, it's not complicated. All right. It's just beyond the scope of this course. So the first step is that those sodium channels are closed. Okay. So there's no ion movement. There's potential, but there's no ion movement. There's then stimulation, right? And that causes an action potential to start. Okay. Once the action potential reaches that closed sodium channel, that stimulates that channel and causes the channel to open. Now, a channel doesn't do anything active. Okay. Everything that a channel does is passive. So the channel opens. Well, the sodium is more concentrated outside than inside. Okay. So it's not only, the sodium is not only drawn in by charge, but it's also drawn in by diffusion. Negative becomes more and more and more positive. Thus, the graph, right? If you put a sensor, an electrochemical sensor on the neuron, you can actually measure, you can quantify this flow of sodium. Here's the negative charge on the neuron, what we call the resting potential, because the neuron isn't doing anything. It's just sitting at rest with the potential to have ions flow, okay? There's then, ready? Stimulation. Sodium channels open, sodium rushes in. As the positive ions, the cations, rush in, that's positive flowing into the neuron. That causes the overall neuron charge to become more and more positive. Since the cell is no longer polarized, we call this process depolarization. Once the sodium channels have opened and the maximum amount of sodium has flowed in, you reach a maximum here. Now the sodium channels close, and now it's time for the sodium potassium pump to do its work. Okay, the sodium potassium pump now starts pumping 3 sodium out, 2 potassium in, 3 sodium out, 2 potassium in. You start creating that debt again, and so you start reducing the charge of the neuron. So the overall neuron now starts to become negative again. So we call this repolarization. The sodium potassium pump always pumps in too much sodium, pumps in too much potassium and pumps out too much sodium. So there's a control mechanism here. Again, we're not going to talk about it right now. It's not complicated, it's too much. And so there's a control mechanism here called the overshot, and then you're back to where you started. Okay, and so then this graph of the flow of sodium ions, you're actually very familiar with. That's the ECG or the EKG that we talked about a couple of topics ago. I think it's topic 1 or 2 when we were talking about the heart. And when someone sits in the hospital and they have the electrodes on their chest, those are the electrodes that I'm talking about. As the sodium ions flow in and out of the neurons in their heart, we can actually measure or quantify the flow of those cations. That's where the EKG comes from. And so here you have depolarization, repolarization, depolarization, repolarization, overshot. And so when I first introduced the EKG, and there's a lot of biochemistry here, right? This is the action of that. Okay, this is a beautiful core that it all starts to really tie together. The only drawback is you got to reach back to core 1, question 4C, isn't it? Yeah, core 1, question 4C, and reach back to that core. And please, please, please link all this together. I would strongly suggest that you start writing out step 1, step 2, step 3, step 4. What makes this content complicated is there are so many steps, right? So good luck. So break down those steps and understand that depolarization here is a 5-step process. And once you learn the 5 steps of the story, it becomes a lot more clear. But to sit back and watch me do it and go, oh, that makes sense, you have to own it. Because what IB does on these exams is they ask you the classic illustrate and explain, diagram and discuss, draw a picture of this and talk about the process. Outline the steps of depolarization in a neuron. Include diagrams of support. All right, draw a picture and talk about it. Okay, so as these neurons then depolarize, I guess I'll throw it on the floor. Come back here. As these neurons depolarize and they carry impulses, I'm looking for a specific image that I was actually labeling just a minute ago. There it is. So what we talked about with the chemistry that happens right in there on the axon of a neuron. But you have to understand that if I need an impulse to go from my brain out to my body, that impulse has to go between multiple neurons. Okay, so where one neuron stops, the terminal, okay, and where the next neuron starts, the dendrite, there's a gap. All right, that's what we're trying to draw here. And so that gap is called the synapse. And the action potential, I should add that to this diagram. The action potential does this down this series of nerves. Okay, so the signal has to be transmitted across this gap. There has to be what we call transmission. Thus, the message being transferred across that gap we call synaptic transmission. It's a fancy term. There's a lot of fancy terminology here. Just think about what the words mean. So transmission means to go from point A to point B. Synaptic means across the synapse, thus you have synaptic transmission. So as with everything else in the neuron, there's chemistry here, okay? Biochemistry has steps. This happens first, and this happens second, and this happens third. Synaptic transmission, that gap right there has 8 steps. Those 8 steps are right there, okay? So that little red circle there, like this little red circle there is what we're drawing down here, okay? This end of the terminal here is the terminal here, okay? And this beginning of the dendrite here is the beginning of the dendrite there. So this is the synapse. This is the gap between 2 neurons when you're taking the action potential from one neuron and trying to get the action potential to start at the next neuron. 8 steps, okay? The action potential arrives, step 1. Step 2, the arrival of the action potential opens calcium gates, right? When calcium gates open, the calcium rushes in. Okay, now something I'm not talking about here is if there's a lot of calcium out here, there has to be some pump that's pumping the calcium out. Okay, IB doesn't hold you accountable to this. So if you guys are bio majors, you'll go to college, professor will stand in front of you, they'll talk about synaptic transmission, and they'll talk to you about the calcium pump that sits right there that pumps out all the calcium. Okay. But when you're teaching neurobiology, there's so much detail that you have to cut some of the detail out to get the concept across, okay? My role at the high school is to be sure that you understand that calcium flows in and that causes the formation of vesicles. How the calcium got there, you don't have to worry about that, right? But you then go to college with this background, and then the professor talks to you about the calcium pump here that through active transport pumps the calcium out. It's not complicated. And then there's just one more layer to your understanding. So action potential arrives, that opens calcium channels. The calcium rushes in. Well, why? There's always a reason why things happen in biology. When the calcium rushes in, that causes the formation of vesicles. All right, so you form vesicles. Same question. Why? Why do you form vesicles? Because inside of these vesicles are proteins called neurotransmitters. We call them neurotransmitters because they transmit the message from one neuron to the next one, right? So then those vesicles contain neurotransmitters. The vesicles then move to the membrane of the terminal. If IB wants to get fancy, they will call this the presynaptic membrane. I don't care for that term. I think it's too fancy. You're just clouding the concept, but they might use it. So this is the membrane of the terminal or the presynaptic membrane. The vesicles then fuse with the terminal membrane, and then it is regular exocytosis. That's core one, right? With exocytosis of the neurotransmitter, since we're focusing on motor neurons, the neurons that control muscle, right? The neurotransmitter for muscle neurons or motor neurons is acetylcholine. And so you write that as capital A, capital C, lowercase h, ACH, right? So the neurotransmitter then diffuses across the synapse, synaptic transmission. The acetylcholine is a key. It's a key that unlocks sodium gates or sodium channels, all right? The sodium channels usually are closed. Once the neurotransmitter diffuses across, bonds to the sodium channel opens. Sodium channel opens, sodium rushes in. Once the sodium rushes in, that depolarizes the second neuron, and that's the creation of an action potential. You just transmitted the action potential from this neuron to this one in 7 steps. Okay? Now, after that channel is open and the action potential propagates away, you've got to close that sodium channel so that the sodium potassium pump can repolarize this area of the neuron. Well, how do you close a door? You got to take the key out of the lock. So if you take the key out, how do you get rid of this protein? You digest it, right? How do you digest it? With an esterase. So acetylcholine esterase. Okay, and I don't think I ever wrote that out, and I should. Acetylcholine esterase. Can't keep track of my pens this morning. There it is. Acetylcholine esterase is an enzyme that digests acetylcholine. So it's acetylcholine esterase. Okay. ACE for the enzyme. Ester is a type of bond. If you want to know ester bond, let me think about that. Yeah, there'll be 2 bonds. Doesn't matter what it is though. It's like that. It's like such. It's 2 carbons. IB will never ask you this. It's 2 carbons with another reference group over here and a double bonded O. That's called an ester bond. Extremely common in all biological molecules. And so if you want to digest just about any biological molecule, you're going to use an esterase. And so the esterase that digests acetylcholine is called acetylcholine esterase. This is the enzyme that digests the acetylcholine. The acetylcholine was the key that opened the channel. You digest the key, the channel closes. Once the channel closes, now the sodium potassium pump can repolarize this. Okay, and then you're ready for the next action potential. So this entire process, these 8 steps, literally happen in about 4 to 5 milliseconds, 4 to 5 thousandths of a second, right? By the time you think, oh, I should move my hand, and you move your hand, okay, that impulse has gone across multiple synapses. So if you think how fast you can move your hand, it's not like you think, oh, I should move my hand. 4, 5, and then your hand starts to move. Okay, you think it, and your hand moves almost immediately. It's really not immediate. There's a time difference, but it's really small. Okay, so by the time you think to move your hand moves very soon after you think it. That in that amount of time, the impulse has crossed multiple synapses. This is crazy fast. All right, so take these 8 steps. Really suggest take these 8 steps and just write them out. Right? 1, 2, 3, 4, 5, 6, 7, 8. Describe what happens. Okay. I don't do that for you, and there's a specific reason why. I need you to process this. Okay. I need you to process it through the left-hand side of your brain. When we do something that is this image rich, okay, this is all pictures, pictures, pictures. I'm really stimulating the right-hand side of your brain. We need that information to go through the left-hand side of your brain just as strong. Okay, so if I write things out and you look at it and copy it and write it down, that's still in the right-hand side of your brain. I need you to create it. I need you to process it. That means it'll transfer it over to the left-hand side of your brain and we're teaching both sides of your brain at the same time. Okay, I'm not holding back on you at all. I am actually helping you understand and process and get a depth of understanding of those steps. Okay, there's a lot of detail here that's unfamiliar. All right, but don't be intimidated by it. Just because it's unfamiliar doesn't make it difficult. It makes it unfamiliar. So how do you get familiar with it? You go through it, and go through it, and go through it. That's how you learn biochemistry. You break it down into steps, and you go through those steps multiple times. And all of a sudden, acetylcholine and acetylcholinesterase and presynaptic membrane and postsynaptic membrane and vesicles and channels, all of this vocabulary becomes more familiar, and so you get more comfortable with it. All right? It's simply exposure, I promise.
Mr. Monroe,
Yes.
how does this process work when like sleep?
It still works, but there are certain parts of the brain where the neurotransmitters are inhibited, especially in your cerebellum, in the base of your brain. The base of your brain area called the cerebellum is the part of your brain that controls skeletal muscle. All right, that part of your brain is inhibited when you dream, especially when you're in the rapid eye movement REM sleep. All right, because you're highly active in your dream, yet most of us lay in bed and we're not running and jumping and talking. Okay, so how do you prevent the brain from functioning while you dream? There are inhibitors that are released as you dream that block the neurotransmission so that you don't flop around in your sleep. But for those of us that do flop around in our sleep, that's because this process isn't fully inhibited. So there's the chemistry here is there's people have PhDs in this, in that you see now, just as an introduction, how much chemistry is here. And biology is the study of what happens when everything works right? But disease is when all of this chemistry or part of this chemistry doesn't work. Okay, that's what disease is. Disease is when the biology doesn't work properly. Okay, so whether you're talking about Alzheimer's, you're talking about Parkinson's, you're talking about palsy, you're talking about seizures, strokes, what would that be? Epilepsy, schizophrenia, all of these, depending on the label, syndromes, diseases, disorders, is when this chemistry doesn't work properly in a specific part of the brain. And so then you figure out, well, is it because a pump doesn't work, a channel doesn't work, a neurotransmitter doesn't work, an enzyme doesn't work? Okay, have you ever heard of nerve gas? The military uses it. It's nasty stuff. Okay, nerve gas is extremely simple. Nerve gas inhibits acetylcholinesterase. Okay, so what happens is the acetylcholine stimulates this nerve and the sodium channel stays open. That goes to the muscle contracts. You inhibit this enzyme doesn't work. The acetylcholine is not digested. The muscle stays contracted. Okay, you spray this on troops. The troops breathe in. What muscle controls your breathing? The diaphragm. Diaphragm's made of skeletal muscle. So if you expose someone to nerve gas, they breathe in that nerve gas, they actually take a breath, their diaphragm contracts, and usually acetylcholinesterase digests the acetylcholine, the impulse gets cut off, and their diaphragm relaxes. You get exposed to nerve gas, you take a breath, your diaphragm contracts, acetylcholine is inhibited, and your diaphragm stays contracted. You can't breathe out. If you can't breathe out, you can't breathe, and you suffocate. So what people talk about, nerve gas is complicated. Nerve gas is ridiculously simple. It inhibits one enzyme, that one. So how do you treat exposure to nerve gas? You go in under the rib cage with a syringe that contains acetylcholinesterase, and you inject this enzyme directly into the diaphragm, digests the acetylcholine, the diaphragm relaxes, the person can breathe, and they can, or breathe out, exhale, and then they can breathe again. They get oxygen for cell rest. It's all chemistry.
Mr. Manor?
Yeah.
I had a question about seizures.
About seniors?
Seizures.
Oh, seizures. Oh, okay.
So, I have seizures, and when I went to the to my doctor, he said that
it's really hard to find a cure for seizures, and it's really hard to find out what's causing them. And my parents and I were really confused about how you couldn't pinpoint where it was happening. Is it because we have so much movement happening in our brain?
Yes, and it's because there's so many things that can cause a seizure. Seizures can be an issue with your channels, with your neurotransmitters, with the neurons themselves, with the pH and the biochemistry of the fluid in the brain, with your anatomical connection. There's a structure in the very core of the brain that connects the right-hand side to the left-hand side. I can't think of that structure, but if that structure isn't properly formed, that can cause seizures. It's as if someone says, what causes a sneeze? How many different things could cause a sneeze? Dust, or pollen, or bacterial infection, or a viral infection, or black pepper. There's all these different items that can cause a sneeze. That's similar to what can cause a seizure, since there are so many possibilities. Seizure, here comes the term, seizure disorders. I don't like that term. The condition of having seizures is extraordinarily hard to diagnose because there's so many things that can cause a seizure. It's actually more simple if you're diagnosed with epilepsy, even though that's a very serious diagnosis. There are specific treatments for the epilepsy, but not every seizure, as you well know now, is the result of epilepsy. So there are times that they will explain what the seizure does, how to prepare for it, and how to handle it when you have a seizure, how to train the people around you for when you have a seizure, and then really you just kind of live with having an occasional seizure. Has anyone talked to you about like petit mal or grand mal and how to handle that?
I believe so. It's
Okay,
when one of them is bigger than the other, right?
definitely, right. And the symptoms can be, I've had students that will completely black out and they'll fall on the floor and have full body spasms. I've had other students that they just look like they're asleep, but their eyes are open. They'll just, the lights are on, but nobody's home kind of thing. They might breathe heavy. They might hypersalivate. There's all sorts of different symptoms that if you are the individual who is next to that person, you just wait. And so I've had probably 8 to 10 students who've had seizures in class. It's obviously easy to spot, and the biggest thing is you make sure that they don't fall. Especially in my classroom, everything is a hard surface. The desk is a hard surface. The lab bench is a hard surface with a sharp corner. The tile floor, right? So my main concern is concussion, a blow to the head, and right. So you just kneel down next to the student and you just wait. And then at some point they might lose consciousness. Then you get them on the floor, or they have there's a name for it when their eyes stay open, catatonic, where their eyes stay open but they're not conscious. And then you'll see them blink rapidly, and then all of a sudden it's like they come back on, and they're usually very confused, disoriented, and then they start looking around, and you just sit right next to them and just very calmly you go, "Hi, welcome back." And then as a teen, they're really embarrassed. And it's like, no, this doesn't bother me at all, right? And then during the seizure, what am I doing? I'm talking to the rest of the class. Hey guys, so-and-so is having a seizure. It's nothing to be afraid of. Their brain's going to turn back on in just 1 second. We're going to be really chill. We're going to be really calm. We're going to help them out. I need somebody to call the nurse, right? Because we're going to need the golf cart. And it is a structure of support, because seizures are really hard to diagnose and treat. So there's a nice long-winded answer to your question, but it's true.
Thank you.
Yeah, you just be sure that when you go away to college, be sure that you talk to your roommates about your seizures, and be sure that you talk to your professors about your seizures so they understand if you have an event during class or in the dorm. Okay, because someone having a response, someone who is overdosing on drugs, someone who is having anaphylactic shock, someone who is responding to poison, not that drugs aren't poison, but go with me, and someone who is having a grand mal seizure, all look the same physically. Okay, so we don't want them to misunderstand your seizure as you possibly overdosing or a toxin or having some other physiological event. They will know that you're having a seizure and they will know how to treat you. So be sure and tell people. Okay.
Okay.
It's super important. It's easy to help you. It's easy to support you. Just we have to know.
Mhm. Thank
Okay.
you.
You're welcome. Of course. Thanks for sharing. All right. So we then need to talk about hormones, which is topic 6, if memory serves. Yes. So when most people talk about hormones, they don't know the system that produces the hormones. Okay. And so the system that produces hormones is called the endocrine system. And it is one of the least talked about systems in the human body. To me, what cell resp is to biochemistry is what the endocrine system is to human anatomy and physiology, right? In human A&P, everybody wants to talk about brain and heart and lungs and muscles and bones. I totally get that. At the same time, this system is super important. So the endocrine system, what does this do? This functions in the control of somatic cells, body cells, and how the somatic cells are controlled is by messages. And the fancy term for a chemical message is a hormone. That's all a hormone is. So control of somatic cells by sending chemical messages. And these chemical messages are hormones. See, we talk to people all about, oh, middle school and high school and these teens and hormones and blah, blah, blah. But most people don't understand what hormones are, right? All a hormone is it's a chemical message, okay? If I am a cell and I want to communicate with you and you're a cell and you want to communicate with me, we would send messages back and forth. Now, between human beings, you have text or phone call, you talk to each other, that's all communication. Cells can't do that. So how do cells send a text to each other? How do cells talk to each other? They do it chemically, right? And those chemical messages are hormones. That's all hormones are, is one cell producing a product and that product then changing the behavior of another cell. That's all it is. Okay? So yes.
What are the difference between hormones and neurotransmitters?
What is the difference between okay? Oh, got it. That's a cool question. So it's the size of the term. Hormone is the very broad term. Neurotransmitter is a specific term. Okay, so neurotransmitters are a type of hormone. Okay, so all neurotransmitters are hormones. I'll go for that for the most part. All neurotransmitters are hormones, but not all hormones are neurotransmitters. Something like testosterone and estrogen. These are classic hormones, but they're not neurotransmitters. Cool?
Mm-hmm.
Okay. All right. So there are structures, glands that produce these hormones. And there are 5 of them. 1, 2, 3, 4, 5. There are 5 glands to the endocrine system. If this interests you, people that study the endocrine system are endocrinologists. They are highly, highly, highly specialized. The most popular gland that people tend to talk about, especially with teenagers, is the pituitary gland. This gland is quite small. It's about the size of the last digit on your pinky and sits in the very center of your brain. And when this gland produces hormones in very high doses, this is what converts a child to an adult. This is what makes a child sexually capable of reproducing, thus an adult. Okay. Then there are glands in your neck, the thyroid. These are about the size of your whole pinky and sit on each side of your trachea. The thyroid sits just above the collarbone or just above the clavicle here, right? And the thyroid, there's 2 of them, one on each side. It's right here. And those are more in the neck, the upper torso. Then your adrenal glands. Most of you are familiar with these, especially if you're roller coaster riders, right? That rush of adrenaline, where's that adrenaline come from? From your adrenal glands, okay? And your adrenal glands sit on top of your kidneys. In your slightly below your mid back, your kidneys sit right here. Kidneys aren't very big. Kidneys are only about this big, and the adrenal glands sit right on top. The other gland are your gonads. Now, of course, there's a difference in the gonads of males versus females. So the female gonad is the ovary, and the male gonad is the testy. Whether you say testy or testicle and preference, whatever you want. And then we have the pancreas. We discussed this back when we hit the digestive system. And you go, hold it. The pancreas not only functions in the digestive system, but also in the endocrine system? Yes, because the pancreas produces insulin and glucagon, which we'll talk about on Tuesday. And so this is in the abdomen. So what we will do in course 6, starting on course 6, hello, topic 6, starting on Tuesday, is that we simply need to go through some of these glands and some of the hormones, especially insulin and glucagon from the pancreas. IB loves to ask you that question, and we will talk about how these hormones work. So don't forget about topic 1 and 2 on Tuesday, and then I'll go back and I'll take pictures today of topic 5. There's a lot of detail there, and make sure that you have the notes for topic 5. So it'll be topics 1 and 2 on Tuesday, and then topics 3 and 4 the Tuesday after that, and we'll be done with course 6, couple 3 weeks. So if you go trick-or-treating tomorrow night, of course, be safe and social distance, please, 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 social distance. Right? Halloween is awesome. You know that the IB teachers, we usually deck it out for Halloween. And this is the first year that we haven't done Halloween in probably 15 years. I miss it. At the same time, just have fun, but be safe. I sound like such a parent. And that's